This is an examination of the hidden human condition.
This is the Hidden Killers Podcast.
The Hidden Killers Podcast.
With Tony Bruschi.
We're going to break down a whole bunch of testimony in the trial of Lori Vallow Daybell. Of course, you can listen to the raw, unfiltered 4 hours in the 4 segments that we post for you. But we're going to break down some of the testimony piece by piece for you in case you just want to hear specific testimony. What you're about to hear is ISP forensic lab expert Rylene Nowlin testifying in the Lori Vallow Daybell trial. Let's go to the courtroom.
All right, now that the witness is sworn, let me just inquire before we start with the examination. Have you previously viewed any of the trial testimony in this case, listened to it, or read about it?
I have not.
Okay, thank you for that. Please respond to any questions verbally so we can keep a clear record. Please try to avoid speaking at the same time as anyone that's questioning you, and talk directly into that microphone so we get a good record. And with that in mind, Mr. Wood, if you'd like to inquire on direct, you may.
Thank you. Can you state your name and spell your last name for the record?
Rylene, Rylene, Nowlin, Nowlin.
Thank you. Where are you employed?
I am employed by the Idaho State Police Forensic Services Laboratory in Meridian, Idaho.
In what capacity are you employed?
I am the Forensic Laboratory Manager.
Could you please tell the jury your educational background and related work experience that brought you to the position you have now?
I have a Bachelor of Science from the College of Idaho. I've completed hundreds of hours of training in biology screening as well as DNA analysis. I began my career in forensics with the Idaho State Police Forensic Services Laboratory in 2002. I was a bench analyst or an everyday scientist in the biology DNA unit until my promotion to laboratory manager in 2014. My duties included analyzing items of evidence for the presence of body fluids, as well as processing items of evidence for a DNA profile. I was also qualified in the DNA database unit, which is analyzing samples from convicted offenders for upload to the National DNA database. Since promoting to lab manager, I have retained my qualifications in the DNA unit. My duties as lab manager include overseeing the day-to-day operations of the laboratory. I supervise the heads of each department in the laboratory, and I oversee our evidence unit.
Thank you. Do you belong to any professional organizations?
Yes.
Which ones?
I am a member of the Northwest Association of Forensic Scientists, the American Academy of Forensic Sciences, the Association of Crime Laboratory Directors, the American Society of Crime Laboratory Directors. I'm also a molecular biology fellow with the American Board of Criminalistics.
Okay. And have you testified as an expert regarding DNA analysis previously?
Yes.
Approximately how many times?
Approximately 85 times prior to today.
Okay. And have you done, how many times have you tested DNA?
I would say over the course of 20 years, I've tested thousands of samples.
Okay. Let's talk about DNA for a minute. At a 101 level, what is DNA?
DNA is a substance that's found in the cells of our body, and it acts as a blueprint. It tells our bodies not only what form to take, but how to function.
Okay, and so do we all have different DNA?
Between individuals, with the exception of identical twins, our DNA is over 99% the same. And in forensics, we're targeting that less than 1% that's unique to an individual, again, with the exception of identical twins.
Okay, so for any two people in this room, their DNA is about 99% the same.
That's correct. That's why we all have the same basic form and our bodies function in the same way.
Okay. Which body fluids contain DNA?
DNA is found in the cells in our body. So, for instance, in our saliva, it's our cheek cells that are sloughed off into that saliva. In our sweat, it's our skin cells that are sloughed off into that. DNA is also found in our bones and teeth as well.
Okay. And how can DNA testing be used in forensics?
In forensics, again, we're looking at those portions of DNA that are unique to an individual. They don't tell me anything about an individual. I can't tell you what color eyes they have, what color hair, how tall they are. But I can use that profile that's generated from an item of evidence and compare it to known individuals in a case to determine the source.
Okay. And what do you mean by profile?
A DNA profile is developed in our laboratory. We look at multiple different locations on the DNA, and half of our DNA comes from our mom and half from our father. And in looking at those, you receive one variable from each parent. So if I can compare it to blood typing, it's a letter. So we're looking at did you receive an A from one parent and a B from another parent, so you're an AB? This is the same, except we're looking at many more locations, and instead of a letter, it's a number.
Okay. And what is the process to determine who a forensic sample originated from?
It's a multi-step process. The first step, as I mentioned, DNA is found in our cells. So the first step is to break open those cells and pull the DNA out, and then remove all of the other cellular components from that sample. The next step is a process for determining how much DNA is present in that sample. Once that's determined, because in forensics we are typically looking at very low amounts of DNA, the next step is called molecular xeroxing. And it's similar to you have a whole dictionary, but you only want to look at 24 pages in that dictionary. So you're just going to copy those particular pages. Those are those specific locations on the DNA of interest in forensics. And it copies them 1,000,000-fold so that there is enough present to visualize. It's then put on an instrument called a genetic analyzer, and that is what interprets the DNA and gives those number assignments at each of the locations.
And those number assignments at each of those locations, is that called a DNA profile?
We refer to that as a DNA profile, yes.
Okay. So once you generate a DNA profile, what can you do with that? Can you compare it to other DNA profiles?
Yes, it can be compared to other profiles if there was DNA on multiple items of evidence in a case. And it can also be compared to known samples from individuals involved in a case.
Okay. Is this method widely accepted by the scientific community?
Yes, it is.
And what steps do you take to ensure the process works correctly?
There are multiple steps that are taken to ensure the process works correctly. There are controls built into the system. So there are samples known as negative controls. Essentially, they go through the same process as all of the other samples, but no DNA is added to those. So at the end, they should be free from DNA. If there's any DNA present in those, then we have to go back and look to see if something happened. There also is a sample of a known DNA profile. So at the end, I can look at that and make sure the correct profile was obtained so that I know the process worked correctly. In addition, we have multiple safeguards built in. So when I go into the laboratory, everything is bleached down, clean paper is put down. I prepare myself by wearing a lab coat, a mask, gloves. My hair is pulled back. Only one item of evidence is ever open at a time, and evidence samples are processed separately in time and space from samples from known individuals.
Okay. Is the Idaho State Police Lab an accredited DNA forensics lab?
Yes, we are.
What accreditation standards do you meet?
We are accredited through the American Association for Laboratory Accreditation, commonly known as A2LA. We are accredited under the ISO 17025, and that's the International Organization for Standards. 17025 are standards specific to laboratories, just in general laboratories. They have a separate set of standards known as R221, and those are the standards specific to forensic laboratories. So we have to be accredited to all of those standards. In addition, because Idaho State Police Forensic Services uploads samples to the National DNA Database, the FBI has established a separate set of standards known as the Quality Assurance Standards for forensic casework laboratories that we also must adhere to and be audited under.
And just, I should have asked you this earlier. Oh, no, I did ask you that earlier. Never mind. Were you assigned to work at case M2020-2233?
Yes, I was assigned to work some items of evidence in that case.
Does this case correspond to the state of Idaho versus Lori Daybell or Lori Vallow?
Yes, it does.
Okay. For your involvement, what items were sent forward for DNA testing in this case?
The items that I was specifically asked to examine were, do you want the laboratory case item number or just a description?
Just a description at this time.
So the first item were biological samples said to be collected from Tylee Ryan, and that consisted of molars and a section of rib. I was also given biological samples said to have been collected from a Joshua J. Vallow, also consisted of molars and a section of rib. I was also given known samples from individuals in the case, specifically Mr. Dennis Trahan, Trahan, as well as Ms. Vallow.
Okay. Let's talk about Tylee Ryan. Did you do any DNA testing regarding Tylee Ryan?
Yes, I did.
What did you do?
I performed DNA analysis of one of the molars that was submitted, and that process involves thoroughly cleaning the exterior of the tooth to remove any surface contaminants or bacteria that could be present, and then taking that tooth and creating it a pulverized powder, and then that powder is what went forward through that multi-step DNA process that I explained earlier to generate a DNA profile from that item.
Okay. And were you able to generate a profile from that item?
Yes, I was.
And did you do any comparisons between that DNA profile and any other known DNA profiles?
I did.
Who did you compare it against?
I compared it to a DNA profile generated from the sample from Lori Vallow.
Did you write a report detailing the results of that testing?
Yes, I did.
Okay. What were those results?
The comparison I performed was, sorry, that's echoing a little bit, was to determine if Lori Vallow could be a possible parent to the deceased individual said to be Tylee Ryan. So I calculated a statistic. It's known as the single parentage index. And what I determined was that Lori Vallow could not be excluded from being the biological mother of the deceased individual said to be Tylee Ryan. The single parentage index, which is a statistical calculation for the locations of DNA that I examined, was 2,528,000,000, and that 99.9999% of the female population would be expected to be excluded from being a biological mother of the deceased individual said to be Tylee Ryan.
Okay. Is there a way to get to 100% of excluding every other woman as being the mother of Tylee Ryan?
In paternity statistics and paternity calculations, I'm basing it on a population database. And that number I gave, that 2 billion number, that's a likelihood ratio. So it's comparing the two hypotheses. So the first hypothesis is Lori Vallow is the biological mother of the deceased individual. And the second hypothesis is she's not. So that's calculating how likely are those two hypotheses. And the only way to get to 100%, because you're only looking at one half of an individual's DNA, would be to test every woman in the world and compare to that sample.
Okay. But can you just state again for the record what number of women, the percentage, are excluded from being the mother of Tylee Ryan?
Based on the locations of DNA that were analyzed, it's expected that 99.9999% of women would be excluded from being the biological mother of the deceased individual said to be Tylee Ryan.
And what was the statistical likelihood that Lori Vallow is the mother of in this DNA testing of Tylee Ryan?
So to state that differently, it is 2,528,000,000 times more likely that Lori Vallow is the mother of the deceased individual as opposed to another randomly selected woman from the population being the mother.
Okay. And I realized the way I was asking that question is probably not totally appropriate because we're talking about samples, correct? And so that was based on the known sample of Lori Vallow and then the sample from the molar of what was assumed to be Tylee Ryan, correct?
Correct. I did a comparison of those two items.
Did you do any DNA testing related to J.J. Vallow?
I did. I had the tooth sample said to be from Joshua J. Vallow.
Was there anything remarkable about that tooth sample?
The tooth sample was different than any I have seen previously in that when I broke open the tooth, there were still, I guess the best word to say is plump blood vessels still present inside the tooth.
Okay. And what steps did you take in that DNA testing?
I performed the same process as described earlier. The only difference is I had to use forceps, fancy name for tweezers, to peel those blood vessels out because bones are treated differently than other types of tissue, so I needed to separate those and process them differently.
Okay. And how did you process both those items?
I did.
And were you able to generate a DNA profile for those items?
I was.
And between the molar and those vessels, were those profiles identical?
They were.
Okay. Did you compare that sample against any other known samples?
I did. That sample was compared against a sample from a Mr. Dennis Trahan.
Okay. And what were the results of that comparison?
Again, I was looking to see if Mr. Trahan could be a biological parent of the deceased individual. I performed the same statistical calculations, and so I couldn't exclude him from being a biological parent. And for the locations of DNA examined, the single parentage index was 602,000, and the probability of exclusion, other men would be excluded from being the biological father, was 99.9999%.
Okay. And did you put that information in a lab report?
Yes, I did.
Okay. Your Honor, the state has no further questions at this time.
All right. Thank you, Mr. Wood. Any cross-examination?
No, Your Honor.
All right. That will conclude the testimony of this witness then. Is the witness allowed to be excused?
Yes, Your Honor.
No objection.
This is an examination of the hidden human condition.
This is the Hidden Killers Podcast
The Hidden Killers Podcast.
with Tony Bruschi.
That was Rylene Nowlin, ISP forensic lab expert, testifying in the trial of Lori Vallow Daybell. We have more courtroom audio for you in just a moment. Press subscribe so you don't miss any of it. Get an ad-free experience through Apple Podcasts. I'm Tony Bruschi. Stay with us.